So in this video, I want to talk about the light reactions of photosynthesis, which include this idea of the Z-scheme, which we'll get to shortly. And of course, this whole process is photophosphorylation because we're going to make ATP, and that is going to be powered by light. So we're taking water and turning it into oxygen, so that's an oxidation reaction. And then we have NADP plus being reduced to NADPH. That's another thing that we said. So we're going to have this occur in the light reactions. And that, of course, is going to be coupled to the production of ATP. So we have NADP plus being turned into NADPH, and water being turned into, being split into protons and oxygen. That, of course, is coupled to the production of ATP. I'm going through this really quickly because this is just a general overview. This whole thing is accomplished by these two photosystems, Photosystems 2 and Photosystem 1. So what does the overall process look like? So we have Photosystem 2 is where water is split into oxygen and the protons. Where does this happen? This happens in the thylakoid lumen. Okay, and this is important. You'll notice I put this little pink star next to it, and I'll get to that in just a second. This happens in the thylakoid lumen, and the reason why that's important, I'll explain more once we get to the Z-scheme, but keep this in mind for now. So where are the electrons going from and to? The electrons are going, of course, from water, right, initially, to, we'll later see this thing called cytochrome B6F. Okay, so that's really anything in that region is considered photosystem 2, which you might see a little bit differently in the way I draw the Z-scheme, but keep this in mind. So then we have, after photosystem 2, we have this thing called plastocyanin, which is a soluble protein. And what this does is really just, it carries electrons from photosystem 2 to photosystem 1. It's basically the middleman of the entire process. After plastocyanin, of course, comes photosystem 2. Oh, excuse me, photosystem 1. Sorry, photosystem 1. So after plastocyanin, we have photosystem 1. And in photosystem 1, electrons go from this P700, which I'll talk about in just a moment, P700 with a little star next to it, an excited P700, and I'll talk about what this is in just a second, to ferredoxin, which is abbreviated often as FD, and then to flavoprotein, which is FAD, you'll see later. And from there, of course, to NADP plus to make NADPH. Now it's important to know where this happens. This happens in the stroma. And again, I haven't talked about the details of why that's important just yet, but this thing and this thing are important to keep in mind. And all these three things that I've starred here, they all contribute to establishing a particular gradient. That gradient, of course, is the proton gradient or the pH gradient. And I'll talk about more details in just a second once we get to the Z-scheme. So now, what's important to realize is that this overall process, right, is taking this NADP pluses, we're adding a proton to it, and making it, and of course electrons to make it NADPH. How many electrons are we adding to NADP plus in this proton to make NADPH? Well, if I know that NADP plus is going to add a hydrogen, right, in the form of a proton, this overall charge is positive 2, right, from these two positive charges here, and the overall NADPH is neutral. So how many electrons would I have to add in order to make this neutral? Well, 2 electrons, right? 2 negative charges to balance out these 2 positive charges. And water, of course, is oxidized to oxygen and 2 protons, and of course the 2 electrons hop off there. So the overall process again is this. So again, these aren't crazy details that I find too important. What I do want you to realize is that the overall process has a delta G of positive 220 kilojoules per mole, which is a huge positive number. What does that mean? That means that these light reactions are endergonic, right? They require energy. They are not spontaneous. Okay? How is this process driven? If it needs energy, where does that energy come from? Well, from light. Okay, light energy drives this process. How? Let's get to the Z-scheme. So now this is kind of a mess right now, but you'll see what's going to go down in just a second. Okay, so in the Z-scheme, this is what's important to recognize is, first of all, this thing looks like a mess, but I'm going to try my best to make sense of it. If you sort of turn your head to the left, you can kind of see from here, you have this little Z shape going there, there, and there. That's why it's called a Z-scheme. Looks kind of like an N if you're looking at it straight, but whatever. What's supposed to happen? This is all these different things that I've drawn here, right, are complexes along the thylakoid membrane. And that's important to keep in mind. I'll fill in all these little blanks here, and hopefully it'll make a lot of sense. But basically, this Z-scheme is, well, what's happening here is the flow of electrons. So if you notice, I have these gray arrows. All of these gray arrows have a little electron above them. That's basically where the electrons are flowing to. What you should keep in mind is that I've drawn this here, this energy going upwards. So basically, any step that's going upwards is increasing in energy, which means it requires energy. Anything going down, any arrows going down, are steps that are exergonic, that are releasing energy. So in the Z-scheme, these ones going down are exergonic, these ones going up are endergonic. Exergonic, endergonic, exergonic. So let's go through and kind of figure this out. The first thing that we have is we're starting off with water, and we're splitting it into we're oxidizing it to oxygen and, of course, protons. Okay, so now what's important here is that we're releasing these protons into the environment. So this is going to contribute to the proton gradient. Well, where is this happening? Well, what we want to keep in mind is that we want a high concentration on one side and a low concentration on the other side. So if we're taking this water and splitting it, these protons, where are they going? Well, they are going, this is happening in the thylakoid lumen. Okay. Which means that what we're going to be doing here is we're going to be building up a hydrogen ion concentration gradient between the lumen and the stroma, where the thylakoid lumen is supposed to have a high H+ concentration and the stroma will have a low one. So this manganese sort of complex here, what it does is it's often called the oxygen evolving complex. So this manganese complex is called the oxygen evolving complex, which I'm just going to abbreviate as OEC. But basically, it allows oxygen to evolve from water. Okay, so the electrons are given up from water to this manganese protein, and then they go from this manganese protein to this Z complex here, and then eventually they go to this thing, this P680. What this P680 is basically, this is chlorophyll. Okay, so you probably have heard of chlorophyll before when talking about chloroplast. Chlorophyll is a pigment, which is what makes the plant green. And these chlorophylls have particular photosynthetic units. And these photosynthetic units have these things called reaction centers. What's important to get from that is that the light energy reacts at a particular reaction center. There's a particular location within the photosynthetic unit on the chlorophyll that reacts or responds to light. So the reason that I've drawn this yellow arrow here is that this is actually light energy coming in. So this is light energy coming in to this P680. And the reason why it's P680 is because the wavelength of light that's actually coming in is 680 nanometers, which is what this chlorophyll responds to. So why is this light here coming in? Well, I mentioned earlier that any downward electron flow is spontaneous and doesn't require energy, right? But if we're going from P680, once P680 has these electrons, it wants to keep those electrons. It wants to stay reduced. So in order to oxidize it, it's going to require energy. So we have to have an input of energy. That input of energy comes from light. And once we add that light, P680 is in its excited state. So this green star here indicates excited. So now this chlorophyll reaction center carrying those electrons, it's excited. So once we have this excited state. By the way, one thing I didn't mention earlier is that everything bound to this left side of these brackets is typically considered part of Photosystem 2, and anything to the right side of this bracket is considered Photosystem 1. And I will mention though that sometimes this bracket will extend to include cytochrome B6F as part of, and everything before it as part of photosystem 1, because the plastocyanin is often considered the mediator between the two photosystems. Just keep that in mind. Anyway, once this excited P680 photosynthetic unit is excited, the electrons go to phaophytin, and then from phaophytin to plastoquinone, and then from plastoquinone to cytochrome B6F. Now, plastoquinone is interesting because what it does is it takes protons from the stroma and pumps them into the lumen. So here we have protons from the stroma, and they're pumped over to the thylakoid lumen. So notice I wrote this number 2 here. There was a number 1 right here. These things written in these pink numbers here represent the ways in which a proton gradient is established. So plastoquinone pumps protons from the stroma into the thylakoid lumen. So this is the second time we've seen a contribution to the proton gradient. Then what do we have going on here? Then we have these electrons continuing to flow down their down this scheme here towards plastocyanin and then to P700. One thing I want to mention before I talk about P700 and further is that notice that anything that you consider between phaophytin and this P700 here, if I asked you which has a higher reduction potential, plastocyanin or phaophytin, well, which one is further down the chain? Plastocyanin, which means plastocyanin wants to get reduced more than phaophytin does, which means that plastocyanin would have a higher reduction potential. So keep that in mind for anything along this chain. Okay, reduction potentials play a role here. So now once we get to this P700, again, this P700 is very much like this P680 in that it does not want to give up its electrons. So in order to cause that to happen, you have to have light energy come in, right? And of course this is P700 because the wavelength of light that this reaction center responds to is 700 nanometers. So then we have this excited P700. So the excited P700 will give up its electrons to ferrodoxin, and then ferrodoxin will give them to this FAD, or this flavoprotein here. And then what this flavoprotein does is it takes NADP plus protons, and of course these electrons here, to make NADPH. So now notice what's going on here is that these protons here are being used up, right? So we're lowering the concentration of H pluses here. Well, where do we want to lower the H plus concentration? In the stroma, right? So we would expect this process to happen in the stroma. So this is the third thing that actually contributes to establishing the proton gradient. Okay, so what do we net here? The overall result here is we have a high concentration of protons in the lumen and a low H plus concentration in the stroma. So this is our net sort of proton gradient here. So that's the Z scheme. Now, notice about the Z scheme is that many of the steps are actually exergonic, right? All these downward arrows, they're exergonic steps. There are only 2 endergonic steps, right? This step here and this step here. But those are so endergonic that the overall process, despite the fact that there are exergonic steps, the entire process overall is endergonic, okay, because of these 2 steps that require light energy. So now let's get another view of what's going on in the chloroplasts. So what's happening really? So we have photosystem 2 here, right, and then plastoquinone, cytochrome B6F, plastocyanin, photosystem 1, ferrodoxin, FAD, the flavoprotein, which is called ferrodoxin NADP plus oxidoreductase because it's oxidizing ferrodoxin, right? It's getting it to lose its electrons. and it's reducing NADP plus to NADPH. So what's happening here? Why do we care about this? So first of all, let me just fill this in here. This is of course light energy coming in at these two different photosystems. But what's happening? We have the establishing of this proton gradient. So we mentioned here that we have in the thylakoid space, or the thylakoid lumen, we have the first thing that contributes it is splitting water, right? That increases the hydrogen ion concentration in the thylakoid space or lumen. The other thing, the second thing, was what happens at plastocyanin. It takes protons from the stroma, right, from the outside, from the stroma, into the thylakoid space, thylakoid lumen. Okay, so then we have this buildup of protons in here. We have a bunch of protons in the thylakoid space, and I can actually go through and just draw a bunch of these things, right? We have a bunch in here. We have a few out here, right? But most of them are, of course, in the thylakoid space, thylakoid lumen. So that's the second thing, right? Plastocyanin is the second thing that contributes to the proton gradient. And of course, the third thing up here, right, is when NADP plus uses up these when NADP plus picks up these protons are now bound to NADP plus to make NADPH, right? So their concentration outside in the stroma is decreasing. So we have this relative difference between the lumen and the stroma, where we have a high concentration of H pluses in here, right, and a low concentration of H pluses out in the stroma. So what happens? In the chloroplast, it's a very similar process to what happens in the electron transport chain. Where do these protons want to go? They want to flow spontaneously down their concentration gradient. So they want to go back out to the stroma, but they can't. They need a particular channel protein, so that will allow them to do that. So that channel protein is ATP synthase. Okay. Now it's a little bit different than the mitochondria's ATP synthase, because there's probably slight differences in structure. There's still a channel protein called the FO, specifically the CFO. C stands for chloroplast. And then, of course, the CF1, which is the actual ATP synthase. So what this does is it couples, right, the flow of these protons out of the lumen back into the stroma, and that spontaneous flow releases energy. That released energy is harnessed to power the phosphorylation of ADP to make ATP. And that's essentially it. So that's pretty much the light reactions. I hope that video was helpful. I do want to mention one quick thing, in case some of the names that I mentioned were a little bit tough to spell or to see. I think I wrote them down over here somewhere. Yeah, so there's a little side notes here. The Phaeophytin is spelled like this. At least I think so. Plastocyanin is spelled like this. PC is plastocyanin. FD is ferrodoxin. So those are just little side notes there. I hope that helps clear things up. And of course, I imagine that you have to pause this video and figure things out as you go along, draw things. But I hope this video was helpful. Thanks for watching.